Dr. John Fisher, who's the chair of bioengineering and is the Fischel Family Distinguished Professor at the University of Maryland. He's the NIBIB NIH Center Director for the Center for Engineering Complex Tissues, and he completed his bachelor's degree in biomedical and chemical engineering from the Johns Hopkins University, master's in chemical engineering at the University of Cincinnati, and his doctoral degree was from Rice, and his postdoctoral fellowship was from University of California, Davis. He has roots in Kentucky. For those that didn't know, his family owns the Willett Distillery, so he's local. It's a small world, and please join me in welcoming him for this seminar titled 3D Printing for Engineering Complex Tissues. Thank you.
Can everybody hear me okay? Yeah. Thank you so much. It's been a really wonderful visit here to Louisville. As was mentioned, I started coming to, my mom's family is from Barts Town, which is about 30 miles away. I started coming to visit my grandparents when I was 6 months old, and I spent a gigantic amount of my childhood here. We usually came to Louisville to go to dinner or shop or something, and went back to Barts Town. But it's really nice to come to visit everyone in Kentucky. And I've had a really wonderful visit yesterday and today, and thank you so much for the hospitality and everything. As was mentioned, my grandfather is, I just thought I'd throw this in here, it's my grandfather, Thompson Willett. He started Willett Distillery about 80 years ago. I remember running as a child and visiting the distillery. It kind of went through a slow period in the 80s, and it's now run by my cousins. My uncle for a while bought bourbon from the surrounding distilleries and blended it, and they made a lot of product in that fashion, and now they're making their own bourbon again. But this is grandfather with his some of his brothers. He had a big family in Barts Town, and their big label at the time when I was a child was called Old Barts Town. That's the name of this racehorse. You can see the painting of the horse here that grandfather's holding, and the horse himself. And this is a picture from the distillery. So any folks actually from Kentucky here? There's got to be some folks from Kentucky. Because I was thinking about, somebody came to Maryland and talked about their connections to Maryland. We'd have about 2 people in our department who are actually from Maryland. But I thought the folks from Kentucky might appreciate this. And if you ever get a chance, if you haven't visited Barts Town, it's a really nice spot to go visit. In addition to all the distilleries, it's a very historic town. I think my uncle's actually mayor right now of Barts Town, so it's a really nice spot. We were just back there a month or 2 ago. My children love to visit my family there. So with that, I also, since this was mentioned, I'm the chair of the department at Maryland. I thought I'd say a few words about the University of Maryland and our department there. Similar to here, we're a relatively new department. We had a graduate program that started in 2002, and then in 2006, Dr. Bob Fischel donated over $30 million to endow the Fischel Department of Bioengineering. So we had the graduate program, we added an undergraduate program, and then built the department from that time. So right now we're about a little bit over 500 undergraduates, so that's somewhere in like the top 10 in terms of size of undergraduate programs. We have about 100 graduate students, the vast majority of which are PhD students. We'd like to see that number continue to grow. We have about 20 faculty members, and we're somewhere around $14 or $15 million in research expenditures. It's really a great spot where we've been fortunate, as you all hear, we've been fortunate to be able to recruit some outstanding young faculty. I just showed 2 here. Kim Stroka, she actually did her PhD with us and did a postdoc at Hopkins, and we were able to recruit her back. And then Greg Duncan is also a Johns Hopkins person. There are some of our new assistant professors, both of which won the Burroughs Wellcome Career Award. I don't think there are too many departments that have 2 professors that won this award. So our joke now is, come to Maryland, you too can win the Burroughs Wellcome Career Award. And then finally, similar to here in Louisville, we just opened up a new building. It's called Clark Hall. You can see the building up on the top left. So we're the Clark School of Engineering. A. James Clark was an alum from the School of Engineering. He was a civil engineering student, went into the construction business, and eventually became president of CEO of, it's big on the East Coast, what's known as Clark Construction. He was a very good friend and benefactor of the School of Engineering, and donated funds, in addition to a lot of things, donated funds to help get this building on track. So the building opened in early 2018. We moved in at that time. It's a 6-story. It's about 180,000 square feet, about $160 million facility. It has floors that are campus, college-wide, and then the department, as well as we have an institute associated with the department, are all housed in this building. So Maryland, we're inside the DC Beltway. We're like right in the northeast corner of the DC Beltway in College Park. If you're ever in DC and want to stop by and say hello, we'd be thrilled to have you come by and visit. I'd be happy to show you around. So getting to my work, as was mentioned, our lab does a lot of work in materials and tissue engineering, and a lot of printing work that I'll talk about for a minute. But just as a kind of introduction, when you think about the history of materials and the concepts that were driving materials developments over the decades, first folks were really thinking about materials and the idea that we wanted to plant something that was relatively inert to the surrounding biological system. Over time, people thought, no, maybe we want materials that have more activity, that respond, and then ultimately materials that have some regenerative properties. So this has been driving a lot of work in tissue engineering, but the question is how do we take polymers, metals, ceramics, and how do we form them into useful three-dimensional biomedical devices? So our lab and many labs have used additive manufacturing approaches to build implantable biomaterial-based products. As I learned quite a bit during my time here, folks here do a lot of work in printing technologies, light-based printing, powder-based printing, extrusion printing, for a variety of applications. So in dentistry, the work in tissue models has been going on for years and years. Surgery, medical devices, even more recently things building drugs and drug formulations using printing approaches. And the printers themselves have gotten vastly less expensive, and that's really helped spur the explosion of printing and bioprinting in the field. So now it's grown considerably over the last few years. Application in healthcare is the third largest sector in additive manufacturing. So if you focus on the bio side of things, probably the largest type of printing approaches that are utilized include lithographic-based approaches, inkjet, laser sintering, extrusion-based printing, and laminate manufacturing. Our lab does most of its work in lithographic as well as extrusion-based printing, and I'm sure many folks are familiar here, but I just thought I'd say a few words from our point of view about these printing approaches and techniques. And I have the obligatory movie on the side here that you can watch while I'm talking. So this is a lithography-based printing approach where you have a photocurable polymer resin. A plane of light is shined up through the bottom of that resin and cures the resin into thin layers repeatedly over time to build a three-dimensional object. Since it's light-based, it has relatively good precision, somewhere on the order of about 20 microns. And we can also use the light as a way to control the cross-linking and therefore the mechanical properties or perhaps even the decorative properties of material. The fabrication is slow, so this little movie that I just showed, I think it takes about 6 or 7 hours to build that little model heart. And as you also saw in the movie, it often requires the building of support structures for that object that you're creating. The materials you can use are pretty vast, but they do need to be photocurable, so you can use things like modified PEG or different polyesters. We've also been doing a lot of work where we isolate ECM from tissues and use that as the basis of a bioink in the lab. It should be noted though, if you're working with a polymer, some kind of polymer system, maybe you have a cross-linking monomer, you need to add some photoinitiator to take that light energy and probably initiate radical polymerization. That all makes sense. You probably also need to add at least one, if not two, photoinhibitors, so you prevent diffusive cross-linking. So essentially you're cross-linking this pixel here, but not the adjacent pixel, so you can get good precision in your objects. And the reason I kind of mention this is if you're using a polymer and a cross-linking monomer, initiator, and maybe two inhibitors, you now have a relatively complex system that you need to figure out. So it does take some time if you're developing a new ink, a new polymer for printing. It does take some time to develop it for your specific application. As I mentioned to a couple folks during my visit, we often have people come to our lab and say, I want to print this material. And we're like, yes, we're excited to work with you and help you on that, but it might take some time to develop the chemistry in the system. Oftentimes you may need to think about post-fabrication processing of whatever you've created. As I mentioned, Maryland's inside the DC Beltway. Just a couple miles away is the FDA campus. So we have a lot of interactions with FDA. We have students that spend all their time or half their time at FDA. And what may not be fully appreciated is FDA, particularly CDRH, Center for Devices and Radiological Health, they have basic research laboratories that help them in their regulatory mission. So we've been fortunate to have a number of projects in collaboration with investigators at FDA. And one question that they were interested in looking at is that if we use a lithographic-based printing approach and we print an object in two different orientations, we're obviously going to get a very different lamellar structure. How does that impact the mechanical properties of whatever we build? So we created some modular objects with them, and we're able to identify some general characteristics that one may think about when you're building objects using lithographic printing. Doing these kinds of studies and other work in the lab, we've tried to build some decision tree analyses that help folks, including ourselves, decide in this case what material to use in a particular printing application. So depending upon whether you're interested in cytocompatibility during printing, what cross-linking method, what temperature, and what kind of strength, those are kind of your decision points. We suggest different materials that may be best, at least as a good starting point in your given application. So in addition to lithographic-based printing, we do a lot of extrusion printing. And again, we have the obligatory movie here. This movie is manic, to say the least. It's printing very fast. We have a woman who's doing a project, I'm not going to talk about it today, on building the nipple areolar complex for women who've had complete vasectomies. But you see she's printing multiple materials. Some are cell-based, and also using a light-based cross-linking approach to cure the material at different layers. The extrusion-based printing approach is very nice because it really allows for cell-based printing. The resolution, since you are extruding fibers, is a little bit less, probably on the order, probably starts at about 100 microns and goes up from there. But you can incorporate a viable, functional, healthy cell population. The fabrication is fast. It's not that fast. it's on the order of maybe 10 minutes, not 10 seconds. And again, you have a variety of materials that you can choose from. Probably our workhorse material is gelatin methacrylate, is a basic bio-ink. But again, we use a lot of ECM. We do everything from fibrin to collagen to a lot of synthetic polymers as well. One thing you do need to keep in mind is that if you're extruding a material and that material encapsulates a cell population, you're probably imparting significant shear forces on that cell population. So you need to have some assessment of the impact of that shear, not only simply on the viability, but also the ultimate functionality of that cell population as you're engineering that into your tissue model or a tissue construct or whatever the case may be. So similarly, we've done some studies to characterize these approaches in extrusion-based printing. And this study, we looked at just simple extrusion printing of PLGA. We looked at it at a variety of chemistries, so copolymer ratio, molecular weight, in-cap chemistry, as well as printing conditions, pressure, temperature, speed, and pattern. And we did this for multiple needle diameters and just asked the simple question of what's the fidelity of what we're getting when we print an object compared to the design of that object in CAD. Basically, what's the precision with which we can print the material. In this study, we were able to show that at smaller needle diameters, some chemistry components, things like the copolymer ratio and the molecular weight, do become significant determinants of printing precision when opposed to larger needle diameters. Here we think the explanation for this is, if we all remember Hagen-Poiseuille flow, resistance to flow varies with the fourth power of radius. So basically, as you get to smaller and smaller needle diameters, the resistance to flow or extrusion of that material goes up quite a bit. And similarly, using these approaches, we've created some decision trees on how to choose the best bioprinter for a given application based on the resolution you're willing to work with, how long you're willing to print a material, as well as the cost of the printer itself. Again, we think these are just some good starting points that help us and hopefully help the field as well. So using these approaches, our lab works with a variety of different systems, tissues. We do some work in the cardiovascular area, bone, cartilage, dermal tissue, including the nipple areolar complex project that I mentioned earlier. We have some work building models of the placenta and some work in using printing approaches to build bioreactors or dynamic cell culture systems. So I'm going to tell 3 or maybe 4 stories that kind of describe some of these projects that are ongoing in the lab, and you can help me with the time. Just tell me if I'm going too long. So the first one is using printing approaches to build bioreactors for engineered bone models. So when you think about bioreactors, they really got going in tissue engineering, regenerative medicine, with the idea that if I'm going to build something of any particular size in the lab, anything over really millimeters in scale, I need to create some dynamic culture system to keep that cell population alive. I can easily mold it or fabricate it, I can even print it, but if I'm not delivering nutrients, oxygen at a steady rate to that cell population, it's just not going to survive by passive diffusion through media and through the construct. So the idea that we needed to build a bioreactor for dynamic culture has been out there for quite a bit of time. We also think that bioreactors can be viewed as a way to control the environment that cells and tissues and engineered tissues are growing in. So we can use a bioreactor to control the shear environment, perhaps control cell-to-cell interactions, and even the formation of 3D tissues, going from 2D planar tissues to 3D tissues. So the original idea of this work was how could we move from the PDMS systems, the stamp systems that are kind of ubiquitous in the field, to something where we could use printing and the precision of printing to create complex culture systems. The kind of first generation of this was just a simple flow chamber that we can print quite easily, and in the flow chamber we have an array of columns. The columns impart some shear on the fluid as it's passing through the flow chamber, and obviously we can control the diameter of the columns, the spacing, the offset of the columns, so we can get different shear environments, be it homogeneous environments within the flow chamber or heterogeneous environments. So we can print these constructs like you see here and then use them to ask some interesting biological questions. And I should say, in all of our work, we really try to use printing as a means to either make something that we can't easily make any other way, or to ask, even more importantly probably, or ask a biological question that we can't easily ask any other way. Sometimes in our field, there seems to be a lot of, it's maybe too strong of a word, but gratuitous use of printing, just printing stuff for the sake of printing it. we're very much of the idea that if you can mold something, you should mold it. You should always go with the easiest fabrication approach. But if printing makes sense, by all means do it, but make sure you're using the capabilities of printing to localize materials or localize different cell populations so you can ask some interesting questions. So in this first generation, we were able to create these constructs in CAD using things like SolidWorks. SolidWorks has a very simple CFD module that you can add to it so we can get some good handle on the flow environment and the shear environment within that construct. In this case, we took HMSCs, human mesenchymal stem cells. We added them to the flow chamber and just looked at them initially at day 0 as well as through 7 days of culture. This system's particularly nice. We had built multi-layered systems which work really well, but they're difficult to interrogate. So this single layer system is very nice. We can put it under the microscope and look at things over time. But what we found initially was that when we see the cell population, if you look, they have the tendency to grow at the interface. This is the column in the array. They had the tendency to grow at the interface between the bottom of the column and the substrate itself. Over time, this was dissipated, and you saw it less with higher flows, but at day 0, you really did notice this effect. So this is actually a well-known effect. I wasn't aware of it. The postdoc who was leading this work went back into the literature and was able to find this phenomenon. It's called negative curvature environments. So if you think about different environments, the easiest one is a concave and convex system. In a concave system, the surface area to volume of a cell is smaller in the concave versus the convex. It's metabolically favorable for the cell to live in that environment versus a more spread environment. So in this case, the negative curvature environment provided by the base of the pillar and the substrate is a much more friendly environment for the cell compared to like a positive curvature or a neutral environment. So that explains the phenomena that we see here. And finally, we were able to show that cells liked the flow chamber and lived in it and grew over time in a similar way as they did in static culture. We then went on to look at this cell patterning or localization phenomenon a little bit more deeply. We used something like cytokalasin to interrupt actin polymerization and showed that that interrupted the cell localization next to the base of the pillars to a good extent. And we're able to also demonstrate that if we applied flow for a day, that also created a more homogeneous cell distribution when compared with either static or short times. So then we asked the question, can we use this patterning effect to induce cell differentiation in a robust and also perhaps a patterned way? So we took the human mesenchymal stem cells, we induced their differentiation down the osteoblastic lineage using osteogenic media, and used alkaline phosphatase expression as an early marker of differentiation. And we were able to show that we did see enhanced ALP expression when grown under dynamic culture when compared to the static condition. Then we asked the question, could we correlate this expression of ALP that we see among the cell population to spatial localization within the bioreactor chamber? So Josephine, who did this work, defined this very nicely. It's called a Pearson correlation coefficient. It looks very complicated, but all it essentially is doing is looking at the variance in shear and comparing that to the variance in ALP expression as we assess using quantitative image analysis. So trying to compare these patterns of ALP expression to the patterns in shear. And what was able, a positive correlation, you get a coefficient that's greater than 1. A negative correlation, you get a coefficient that's between 0 and -1. I'm sorry, this is between 0 and 1. And no correlation, you see a coefficient close to 0. And we're able to show that we do indeed see a reasonable correlation when grown under growth media. However, that correlation is dissipated when we use osteogenic media that's driving uniform differentiation among all the cell population. Finally, we went back and using that localization effect, we thought that we could use that to our advantage to create patterns of cell populations within the bioreactor. So if we seed HMSCs first, they like to attach near the base of these pillars. So that leaves the other spaces in the bioreactor available for subsequent cell populations. So we seed something like HUVEX, and we start to get a patterning in our bioreactor system. And that's what's shown here. Distance away from the pillar, it's predominantly HMSCs close to the pillar, but as we get further and further away from the pillar, we see more and more HUVEX. What's interesting is if you switch the seeding order, first HUVEX and then HMSCs, since the HUVEX are a little bit smaller than the HMSCs, that 90-degree angle between the pillar and the substrate is not as favorable to them, so they tend to spread more homogeneously. And then therefore, if you add HMSCs afterwards, you get a homogeneous distribution of the HMSCs. If you were to take that pillar and put it on a little bit of an angle, so you get a smaller angle between it and the substrate, you do indeed get HUVEC localization in that small angle environment. And finally, we just did some calcium signaling studies. You can see them ongoing here of our different culture systems, the stem cells alone, endothelial cells alone, an unmixed co-culture, and a mixed co-culture. And really what we're just trying to show with these studies is that we can use this environment of the bioreactor to create relatively complex cell systems of multiple cell populations and interrogate their response based on localization. So we're able to show that we get differences in oscillating calcium signaling and the number of calcium spikes as a function of these different culture strategies. So ultimately, we think these printed bioreactors have a lot of advantages. We can use the CAD design, the fluid dynamics to create environments that are particularly favorable to the cell population of interest, and then we can tweak them to get, in this case, co-cultures or differentiation patterns that might be helpful in a given application. We've then taken this work and developed it further for specific applications, although that was in the context of bone. This work, we're trying to create some stratified scaffolds to regenerate dermal tissue. So this project was led by a PhD student, Javier Navarro. Javier is originally from Colombia, and he was fortunate to get a Fulbright fellowship to work in our lab. He recently graduated, and he's now doing a postdoc. And Javier made the observation that in the body there are multiple sites where there are tissue types coming together, but they're coming together in a stratified way. Interfacial tissue engineering and gradient tissues have been looked at, led by work by like Helen Liu at Columbia for quite a few years. But building multiple tissues simultaneously that have stratified layers have been investigated less often. So in this case, Javier was trying to build something that replicated the dermal tissue, adipose tissue layers that we see in skin. And the idea was that I wanted to create a culture system that cultured the dermal tissue and adipose tissue separately, but still allowed for molecular communication among these two cell populations. So we did not want to create something where we got a gradient in this reactor chamber, but something that was more stratified, where we had the dermal-like tissue and the adipose-like tissue adjacent to one another, but the cells could still communicate, there still could be transport of molecular cues between the two reactor chambers. Equally, we could culture the reactor chambers in separate media. We could do this in such a way so that the media either mixed over time, or we could do single pass media flows, so we had completely separate media environments over time. So again, using a printing approach, Javier built these bioreactor systems, and you can kind of see it here. It's about yay big. It has two chambers that fit one on top of the other. The scaffold that he was using is just a simple cube-like scaffold with a lattice structure, and then between the two chambers was a separating membrane, and I'll talk about that membrane in a second. So again, we use some CFD approaches to look at flow through the dual chamber bioreactor as well as mixing and the shear environment, establishing that we could indeed flow multiple medias in the two chambers, and the mixing could be regulated by this the porosity of this membrane that we inserted between the two scaffolds and the two chambers. So building that membrane took a little bit of time since we were working in a dermal application, and we had a nice collaboration with a company from North Carolina that did a lot of work in keratin-based materials. We decided to build a keratin membrane. This membrane is photo cross-linked using a printing approach, and the printing chemistry strategy is just based on riboflavin, which is relatively common in the field. So Javier made the observation that based on two key factors, the amount of exposure time, light exposure time, as well as the thickness of the construct, so how thick the membrane was, you essentially could use those to control the amount of energy that was imparted in the cross-linking of that membrane, and then ultimately the stiffness or cross-linking of that membrane. So that's what's described here. So here are the two values. The first value is exposure time. The second value is membrane thickness. And we showed that we could vary, by varying these two parameters, we could vary the amount of energy that was delivered during the cross-linking reaction, and roughly energy then correlated to cross-linking of the keratin by the riboflavin chemistry with these different energy density deliveries. Using this approach, Javier then validated that we could use the membrane to control molecular communication through the membrane and ultimately to a cell population. So first he characterized the swelling of the membrane, but then took the membrane, put it into a transwell, added adipogenic molecules to the top of the transwell, and then human mesenchymal stem cells at the bottom, and using different membrane types and ultimately different porosities or cross-linkings, was able to show that we can get different amounts of lipid expression as a marker of adipogenic differentiation by controlling the membrane type that was used in the transwell. So then, going back, he took these membranes that he's printed and created, put them back into the bioreactor system, asked similar questions about fluid mixing as a function of the porosity of the membranes, and essentially, as if you have a very, very tight membrane, you're not going to get a lot of mixing between the two chambers of the bioreactor, but as the porosity increases, we get more and more mixing. And you see this was computationally done, and this is the experimental result that kind of validates that approach. Finally, getting to the real experimental work, the first kind of test was taking fibroblasts, putting them in both chambers, the upper, lower chamber, so chamber A and B, the same kind of media, and then growing them over time and demonstrating that we get good, healthy, robust fibroblast proliferation and matrix expression in the two chambers throughout a 28-day study. The next study was a little bit more of what we were hoping to do, where he took human mesenchymal stem cells, put them in both chambers in line A and line B, and then after 7 days added adipogenic media, so media that would induce the adipogenic differentiation of the stem cells, put that in the top chamber in line A, and then looked at the differentiation of the stem cells in the bottom chamber in line B. The only way these cells should really be differentiating is because of the molecular transport of these factors from the top chamber down to the bottom chamber. And cultured these for 28 days and was able to demonstrate that he did indeed find, again, as using lipid as a marker, lipid expression in both the top and the bottom chambers for multiple membrane types, and correlating the expression to the membrane cross-linking, or ultimately the membrane porosity. The last thing that Javier was interested in doing was, this was a dermal project, particularly for patients who had facial wounds. there's really interest in trying to create complex dermal structures. So up until this time, the interface between these two chambers was a flat plane, and Javier was interested in creating more complex things where you get some curvature between these two chambers. So the idea was, how could we build in some structural complexity between the two chambers? And he did this using the membrane approach and the shape of the scaffolds that went into the two chambers. So you can see the kind of design here, and then ultimately how it fit together in the bioreactors that he built. So this was kind of an interesting kind of spatial study where he's just using fluid dye to illuminate the bottom chamber, and did this as a function of membrane shape. So here you can see a single continuous scaffold with no membrane separating the upper and lower chamber. This is not the membrane, this is a rubber gasket that separates the two chambers. So here's a single continuous scaffold, and you see the dye diffusing across. Here's two different scaffolds, but with a membrane, a very tight membrane that's preventing diffusion from the bottom chamber up to the top. And here's a membrane where we have, the percentage is done by space, but a 25% curved scaffold that's separating the two chambers. You can start to see some structure in the bottom. The bottom chamber is equally, you can see that in the top chamber. When you extend that curvature, when you make it even more pronounced, you actually start to see some dead zones in the bottom scaffold. It's just the curvature is too much for simple diffusion, a little bit of convection to fill this bottom chamber. So Javier then validated this using a cell, because the previous work is just looking at a dye, and validated this using a cell approach, and again was able to demonstrate the cells survived and proliferated in these complex environments. And then finally did some quantitative analysis, again, of, in this case, using a diptonectin as a marker, and then as a function of these different approaches. Ultimately, we think this is a really exciting approach to use a membrane and a printed bioreactor to control the structure and the growth of tissues that have complexity, not only in their stratified nature, but also the geometry of that stratified nature. We're able to show that we can sustain and grow multiple cell populations adjacent to one another, but still supporting communication across those multiple cell populations. Ultimately, as I mentioned, the application of this work is to build complex dermal structures that we find in the face, but doing it in such a way that we can build in the stratified layers of the dermal tissue itself. So let's see, what time should I've got maybe like 10 minutes?
Yeah, 5 to 10 minutes.
5 to 10 minutes. I'll just tell one more story on the placenta, and then I'll stop there. So I mentioned that we're relatively close to FDA. On the other side of us, down towards DC, about halfway between us and the White House, is the Children's National Medical Center. And we were fortunate to start a collaboration with folks at Children's that looked at placental biology. So just very, very briefly, as we all know, the placenta is the regulator of communication from maternal to fetal tissue, and is made up of a series, essentially you could think of it as a series of resistance to transport from the maternal blood down to the fetal blood. So we have multiple cell types, trophoblast, cytotrophoblast, and ultimately down to endothelial cells. There, for a number of reasons, there aren't really good animal models for the human placenta. So what's often done is either transwell studies or ex vivo culture of the placenta. So taking a term placenta and then perfusing that and working that as a tissue model. It also needs to be emphasized too that a 9-month term placenta does not function in the same way as a 2-month placenta or 4-month placenta. the cell population, the matrix, it's all completely different. So that kind of motivated us to try to create a printed model of the placental tissue. We felt like we can work with materials, cell populations, we can put them together in a relatively precise way. This is something that we can easily build and allow us to ask some questions about placental biology. So our first kind of go at building a model of the placenta is this tri-layer structure that you see here. We took two, they might be somewhat imperfect trophoblast cell lines, but they're known as BWO B30 cells. A monolayer of those were our model for the syncytial trophoblasts at the top of the model. Then a hydrogel that had the same cell type, the same BWO cells distributed through it was the middle layer, and at the bottom is a layer, a monolayer of endothelial cells. In this case, we're using HUVEX. If you take a cross section of that printed model and image it, you see something here, something that looks like this, where here are the endothelial sides on the HUVEX on the endothelial sides, the BWOs on the trophoblast side. You don't see the cytotrophoblasts distributed here, not because they're not here, but just because the cell density is so much lower. So we used this construct. First, we needed to validate it. We looked at questions such as the tightness of the monolayers on the top and bottom of the construct. We looked at this both by imaging as well as looking at resistance using tier approaches to demonstrate that we were getting mostly relatively tight monolayers on the top and the bottom of the construct. We also asked questions about whether the cells themselves were acting as they would natively. So both the BWO cells and the HUVEX, were they expressing key markers like HCG? HCG is a protein that's assayed in pregnancy kits. Progesterone and things like VEGF, and we're able to demonstrate that the cells are excreting factors as they normally would, at least in cell culture. So we use these constructs to ask a variety of different questions. Kind of standard transport questions: how do things move from the maternal side to the fetal side? More complex things: disease transmission, and then finally looking at the effects of medications that moms might be taking during pregnancy and how they might affect fetal development. I should emphasize, we can look at the response of the model placental tissue itself. So we can take, let's say, a prescription drug and look at the response of that cell population, but we can also take that model, put it into a transwell on top of, now this is now a third cell type that's in monolayer, so we can put some like fetal cardiomyocytes at the bottom and ask questions about how drugs might be metabolized by the model placental tissue and how that might affect the fetal cardiomyocytes. And we've done all these studies. So briefly, we're able to demonstrate like transport of things like glucose, which is relatively quick through the model, as well as larger molecules like IgG through the model, and get some idea of diffusion coefficients. In collaboration with a group at George Washington University who are experts in Zika virus, we're able to do some Zika virus studies where we showed that the Zika virus did indeed pass through our placental model, but some agents, things like chloroquine, could inhibit that. Then when we took this placental model, put it on top of neural progenitor cells, we were able to show that the model itself does inhibit Zika virus transport, as you would hope, as you would expect it to do, when compared to just taking that Zika virus and directly adding it to the neural progenitor cells. Finally, we looked at the response of some prescription medications that may be taken, in this case for antidepressant medications that might be taken by moms during pregnancy. And again, looking at responses both of the model itself as well as subsequent cell populations below the model. We're able to show that the model differentially controlled transport of two different drugs, and that the drugs could be taken up by the barrier cells and then slowly released to fetal cells subsequent to that uptake. So I have one other story, but I think I'll stop here. I do want to say that we were very fortunate a couple of years ago to receive a center grant from NIH. The center is called the Center for Engineering Complex Tissues. This center it's a P41 mechanism. So we're doing it in collaborations with folks at Rice as well as folks at Wake Forest. The folks at Rice are building scaffolds with complex structure. The folks at Wake Forest are focusing on cell printing, and we're doing our bioreactor culture, some of the studies that I described earlier. But these centers, the P41 centers, are really meant to build a community around a technology. So there are a number of ways that you folks can interact with us through what are known as either collaborative projects or service projects. The distinction between these two is not so important right now, but ultimately what I'm trying to get to is if there are opportunities to work with anyone here, we'd be very excited to do so. So for like our component, if there are bioreactor systems that we can build for you, if there are ways that we can help you build systems, if folks want to come to our lab and train, we can do that as well. We'd be very excited to explore those opportunities with anyone. So this is just another slide on the center and my email address in case anybody would like to connect with me. And it's called CSTI, and the website's address is right there as well. And there are plenty of information on the website to connect with us. So with that, I'd like to acknowledge the students that work in the lab. So we heard about the bone bioreactor study that was led primarily by Josephine Lembong. Josephine was an outstanding postdoc in our lab. She did her PhD at Princeton and then joined us. She now works for a small company just north of us called Rooster Bio. If you're familiar with Rooster Bio, they are selling MSCs, mesenchymal stem cells, at large scale for clinical and other applications. Then we heard about the placental work that was done by Vincent Kuo as well as Naveen on the placental work, and then the dermal tissue work was done by Javier Navarro. I'd really like to acknowledge all of our financial support, and thank you so much for your attention. I'd be happy to answer any questions that you may have.
So you were using endothelial cells. Have you looked at the cyclic stretch of endothelial cells? Because some of the studies that we've done, the cyclic stretch, the periodic cyclic stretch that goes through, seems to basically alter even the geometry of the endothelial cells. Endothelial cells kind of orient themselves to the flow, so sure, there's shear dependency, but there also seems to be that cyclic stretch dependency where, for example, growth factors like VEGF and release of production of 1 milligram factor and a number of other things are related to cyclic stretch, not just shear. And so you have this, we've been trying to, we've even had a co-culture model where we have smooth muscle cells and endothelial cells to create a vascular model to study this. Clinically, it affects patients with these ventricular assist devices because they don't have a pulse.
Right.
So the cyclic stretches act and they end up with bleeding issues in the intestine.
So we've done a number of studies looking at what's really the best endothelial cell type to start with. HUVEC's not probably the best. So we've done some work on EPCs. We're doing some work with IPS-derived endothelial-like cells now. So we focused on that. But I definitely agree with you. We have some efforts right now to build some of these bioreactor systems that could impart cyclic stretch or a variety of mechanical stimulations to our cultures, and we hope to have those up and running, shortly, because this is something we often get asked, and as I mentioned, it's kind of the role in the center. We'd really like to be able to provide these for our applications, but also for the community as well.
We have those systems, we can talk later.
Yeah, yeah, absolutely, yeah. Yeah.
So when you're talking about 3D printing with like selectively placing different cell types and stuff, do you know if anyone has worked with also having the ability to selectively place like signaling factors or cytokines or anything like that where you're...
Yeah, so I didn't mention it, but we've done that like in our placental studies. We did some trophoblast migration studies. There's this whole story when the placenta is embedded in the uterine wall. Cell populations, the trophoblasts migrate and allow for the expansion of vasculature that ultimately provides increased nutrition as the fetus is developing. So there's a lot of questions about what's driving that trophoblast migration. So what we did was print, we just did a circular disc so that the geometry and the math is simple, but print circular discs where we'd add a growth factor in a particular spot. We're able to track its diffusion out radially, and then therefore the cell migration in towards that spot. We think these applications are great for printing. Put something where we want to and ask a question about it, and we really try to focus on that. Yeah.
So do you do sort of reaction diffusion modeling along with maybe modeling aspects of cellular metabolism to build these systems?
We haven't done a lot of that. That's a good point. We haven't gotten to probably that level of sophistication. We've really focused on fluid environment and shear as kind of our main components. I should mention too, you design things in CAD and then you print them. You can run the flow simulations off your CAD design, or you can take the object, do a micro CT, so you really get a good handle on what you actually printed, not what you hoped you printed, and then do those same studies. We're doing a lot of those questions now as kind of our next iteration, because there is some distinction between what you design and what you ultimately get. Excellent. Well, thank you so much.
So at the end of the seminar, we like to thank, on behalf of the Department of Bioengineering and the Swedish School of Engineering, we like to thank Dr. John Fisher for accepting our invitation and coming to share his research with us. So this is a little small proof of bioengineering.
This is really too much, but thank you so much. Excellent. Thank you so much.
Beautiful.
Oh, okay.
This is small part.
Excellent. Perfect. Thank you. That's beautiful.
So we have lunch, so please feel free to grab you some pizza and drink and go.
Yeah. I know we're short on time, so. I know you're short on time, so. I'm in electrical engineering. I'm working on polymer